Welcome back to the channel. Today we'll be looking at a lecture on plant propagation slash reproduction. So both terms mean the same. So propagation and reproduction, they are used interchangeable and they mean the same. However, please note that we will be talking about plant propagation today. There is also animal reproduction, but we will not be looking at that type of reproduction. Today we will be focusing on the plants. If you have not yet subscribed to the channel, please go ahead right now and hit that subscription button. Let's get straight into the lesson. Objectives. Objective number 1. To define the term plant propagation. Number 2. To name the two types of plant propagation and define them. Number 3. To list the types of vegetative propagation methods, cuttings, layering, budding, grafting, and tissue culture, and define them. 4. To explain the advantages and disadvantages of sexual and asexual reproduction. So remember that all these objectives, they are speaking about the plant, not the animal. What is plant propagation? Plant propagation or reproduction is the process of increasing the number of plants from the parent stock. So in other words, it means to make more or new plants, the multiplication of new plants, the creation of new plants. Types of reproduction. There are two basic types of propagation. These are 1. Sexual propagation. Now what that means is that sex cells are involved in this type of reproduction, and of course the sex cells are produced by the flower of the plant. Number 2, asexual propagation, and this type of propagation is also known as vegetative propagation. Of course, with this type of reproduction, no sex cells are involved, but what are involved are the leaf, the stem, the root, the buds of the plant, but no sex cells. So the function of the flower is not involved in this type of reproduction. Sexual propagation. So let's get more to the sexual side of the plant propagation. What is sexual propagation? Now, this is the creation of new plants by the joining of the male sex cell, which is called the pollen grain, and the female sex cell, which is called the ovule, to produce seeds. So in short, it is the joining of the pollen grain and the ovule that will result in the production of seeds. Seeds will develop into a new generation of plants. So of course what this is saying now, when you plant these seeds will turn into new plants. The flower of the plant produces these seeds. So the part of the plant that is involved in this type of reproduction is the flower of the plant. In the next slide we will look at the parts of the flower just to give you a reminder of what the parts are. And the third point here, the new plants that is produced from a seed is called a seedling. Alright, so please note that in opposite to asexual reproduction, the new plants that are formed, they are referred to as plantlets. But once they are developed from a seed, they are called seedling. Alright, so parts of the flower. And of course, the flower we oftentimes call them blossoms. So we said the plant blossom, the mango tree blossom. So those we are talking about the flower of the plant. Alright, so the flower, of course, as you realize, is that it produced a male sex cell and a female sex cell. Alright, so let's get into the female parts of the flower. So the ovule here is the female sex cell. Alright, so this structure here is the ovule. We have the ovary that produces the ovule. We have the style here and the stigma. The stigma catches the pollen grain. And the style supports the stigma. Alright, all those four parts together make up the female parts of the plant, and that is referred to as a pistil or carpel. Alright, so let's go to the male parts of the flower. You have the pollen grain, which is this black structure here, which is produced by the anther. So you have the anther, the yellow structure that produces the pollen grain, and of course you have the filament that supports, which is this tube here that supports the anther. And the male parts are termed stamen, of course. And there are other parts of the flower that help with the process of the flower. Alright, so that helps the flower to carry out its function. And of course you have the petal, which is the brightly colored part here. These brightly colored structures here. Alright, so Let's move to the advantages of sexual propagation. Alright, advantage number 1. New offsprings are non-identical to the parent plants. So with the advantage with this, you can get new plants being produced at all times. They are different from their parents. So you can get maybe a plant that can resist disease because you have a combination of two plants coming together. So one might be good at resisting disease, one might produce a lot of fruit. So you can get plants that are hybrids, that are vigorous. Alright, advantage number 2, plants produced have strong root system for good anchorage. Okay. 3, transportation of seed is easy. So in terms of transporting seeds compared to transporting plants, grown plants, it would be difficult, but with seeds it would be very easy. So that's a good thing. Number 4, sexual propagation give rise to different varieties. So that's similar to number 1. So for example, you want to produce yellow hibiscus flowers or pink or purple. Well, this is the method to do that. Alright, the disadvantages of sexual propagation. Alright, number 1, offsprings take a long time to grow and fruit. So yes, that's the disadvantage. So when you plant a plant from seed, some plants might take up to 5 years to mature and then start to bear. In other words, in comparing it to the asexual, with those methods, a plant can bear within 3 years. That would normally take like 5 years with growing from seed. Number 2, two parents are required. Alright, so the disadvantage with this method, two parents are required. Number 2, large roots can damage buildings. Alright, so this should be number 3 instead. Large roots can damage buildings. What we have here as number 3, some plant diseases are spread by seeds. So what that is saying is that some plant disease, they live in the seeds of the plant. And when you plant those seeds and the plants start to grow, the disease will spread to the plants. Alright, so let's move to asexual propagation. Asexual propagation is the creation of new plants without the use of sex cells. So of course, new plants are produced without the use of sex cells and also seeds. So it is production of new plants without the use of seeds or sex cells. Alright, the second point, this process involves vegetative parts of the plant. So the vegetative parts of the plant are stem, root, buds, and leaves, which develop to produce a new plant. So I'll explain later how the plant uses the stem, the root, the buds, and leaves to produce new plant. I'll explain that shortly. Alright, so another important point to note is that it is the seedless multiplication of plants. So in some cases, it is referred to as the seedless multiplication of plants, meaning there are no seeds that are involved in this type of reproduction. Alright, so with sexual reproduction, seeds are formed. With asexual reproduction, no seeds are formed. Alright, there are two types of vegetative propagation. So number 1, we have natural vegetative propagation. And with this type of reproduction, it means that man does not involve in this type of reproduction. So that's the difference. No seeds are involved, no man will help the plant in this type of reproduction. So in other words, you plant a banana tree, the banana tree will reproduce on its own. So it will continue to reproduce on its own. So man does not involve in this type of reproduction. Alright, number 2, artificial vegetative propagation. With this type of reproduction, man is involved in the reproduction process of the plants. No seeds are involved, no flowers are involved, but man is involved. Alright, so man will carry out use a technique to induce the plant to reproduce. Alright, so that's the difference between the two. Alright, let's continue. Alright, so let's go into the definition of natural vegetative propagation. Alright, this is the creation of new plants without the use of sex cells. So remember, no sex cells and without the help of man. Okay? Alright, so we have different examples of plants that reproduce this way. So we look at examples of those plants that reproduce naturally, asexually, and also the name of the parts that carry out that type of reproduction. So what I mean is that, for example, the ginger plant, they reproduce naturally, asexually, and the parts that they use to carry out that reproduction is called rhizomes. Alright, so we'll be looking at the examples similar in this way. Alright, so let's look at rhizomes. So a rhizome is found within a ginger and a turmeric plant, for example. Alright, so let's look at the definition of rhizome. So a rhizome is a modified underground stem that runs horizontally under the ground. So the rhizome is normally produced by a ginger, and I soon give you an example of that in a diagram in the next slide. So it is a stem. So the rhizome is a stem, but that stem, it grows under the ground and it is produced by the ginger plant or the turmeric plant, but it runs horizontally under the ground. Now that rhizome is able to reproduce on its own. So I'll explain shortly how the rhizome will reproduce on its own. Alright, B, you have runners or stolon, and these are aerial stems that creep along the ground. For example of these plants that use runners are nutgrass and pimento grass. C, bulbs. Bulbs are very short stem with fleshy foliage, leaves, and buds. The onion and garlic plant normally use bulbs to reproduce, and the bulbs, these are stems, but they are short stems and they have those fleshy leaves that store a lot of food, and it helps the onion and the garlic plant to produce new plants without the use of seeds. D, stem tubers, underground stems with buds and scale leaves. For example, your Irish potato and your yam. So your yam is a stem. Your Irish potato is a stem. It's a stem of a plant, but it's underground. Alright, E, root tuber. These are adventitious roots, but do not contain scale leaves. So the sweet potato and the cassava, those are actually roots. They are root tuber, and they are developed from adventitious roots, not stems. F, carm. Carm is a special tuber which grows erect and have contractile roots. So for example, your dasheen plants, your cocoa plants normally reproduce using carms. And G, suckers, shoots which arise adventurously. For example, your banana, your sugarcane, they use suckers. Alright, so the next question now, which is a big question. Although the plant organs reproduce asexually, so that's what we want to find out now. Our onion uses bulb to reproduce. How does the ginger plant use its rhizome to reproduce? Now, they do so by one, using food they stored. Alright, and they use food that they store for energy. Two, they use buds. Of course, buds are very tiny, or we would say young leaves and shoots of plants. And of course, they use three, hormones. And these are chemicals that trigger the growth process and control the growth process within the plant or of the plant. Let's move to the next slide. So, in this slide now, it is answering the question, how does the ginger plant reproduce asexually? Alright, so I'm going to explain this diagram here. So number one, so normally the ginger that you consume is actually the rhizome. Now it is produced by a plant. So in one, say you have a mature ginger plant here. Alright? Alright, number two, the mature ginger plant produces a ginger rhizome. So eventually the plant will produce an underground and horizontal stem here, which is the ginger. This is the part that you normally consume, this yellow structure here. So it will be produced underground. Now this ginger is stored food. It have a lot of stored food in it. It also have buds on it. Alright, so these areas, it have a lot of buds, which are young leaves and stems on it. Okay. 3, rhizome remains in the soil after the shoot system rots. So what will happen if the rhizome remains under the soil even after the plant rot? What will happen? This ginger will grow into a new plant. Alright, using that store food that it have and the buds that it has, it can, and the hormones in it, it will grow new plants. So what happened now is that at 4, the rhizome produce young ginger plants. So the buds that are here, the store food will produce the energy to grow those buds. So once the rhizome here realize that it needs some shoot system and root system to continue its generation, then what will happen? That hormone will trigger these buds to grow, and that energy will come from the food that is stored in the ginger plant. So that is how a ginger plant will normally reproduce asexually. The other plants, they are similar. That's the same method that they'll use. They'll use their store food and hormones to grow buds and produce roots, etc. Alright, so let's get into the artificial vegetative propagation method. This is the production of new offsprings without the use of seeds, but by the help of man. Alright, so please note that with both natural and artificial vegetative propagation, no seeds production are involved. But with this one, with the natural, man was not involved in the reproduction process. But with the artificial method, man is involved. So this is where the technique of man will be used, and I'll explain all of those. Alright, so also please note that new plant produced from the vegetative parts of the plant parent is called a plantlet. So the new plants that are produced by these methods are called a plantlet. So the methods of vegetative propagation, we have 5 of them. 1. Cuttings. 2. Layering. 3. Budding. 4. Grafting. 5. Tissue culture. So I'll explain all of them. I'll give you definitions. Cuttings. Cuttings are pieces of plants that are removed from plants and then set in an area. Those areas could be your soil, your container, your propagating bin, to develop into a new plant. So with this process, you actually remove plant parts and set them in an area. So you remove them, put them in an area to grow shoots and roots. Alright? So we have different types of cuttings. So the types of cuttings are: number 1, stem cutting. There are 3 types of stem cuttings, namely: A, you have hardwood, and this is taken from mature stems. Alright, so plants like your ackee or grape, you can use hardwood cuttings on that. Of course, you'd use your rooting hormone to help those stems grow roots. Alright, B, you have semi-hardwood. So you have hardwood, semi-hardwood. So the semi-hardwood, these are taken from stems where the bark is not fully developed or partially developed wood. Right, so the bark is not fully developed. Alright, you call it semi-hardwood. Or if the wood is not fully developed also, you call those semi-hardwood stems. Alright, so your fruit plants, your cassava, your sugarcane, those are plants that you can actually take your semi-hardwood cuttings from. Alright, C, you have softwood cutting. So of course you have hardwood, semi-hardwood, and softwood cutting. So the softwood is taken from non-woody bark. For example, your sweet potato. Alright, your begonia, etc. Number 2, you have leaf cuttings, which is taken from your begonia. 3, you also have root cuttings, which is taken from your breadfruit, for example. There are the plants, you can also go ahead and research them. So you have 3 types of cuttings overall. You have the stem cutting, leaf cutting, and root cutting. And of course, you have 3 types of stem cuttings: your hardwood, your semi-hardwood, and your softwood. Alright, so this diagram here explains how a cutting is done. So of course, you will select your plant. So number 1, you select your plant that you want a plant from. Right? So remember now that this will give you an identical plant to the parent. So you have a nice hibiscus plant and you want another plant just looking just like that plant. Then of course you select the plant. Alright, after you have selected the plant, you will select a stem on the plant. So in this case, this is a stem that was selected. So a secateur or a roll cut is used to remove a piece of the stem. So this tool here is your secateur or your roll cut, which is like a scissors, and it will be used to take the cuttings off the plant. Alright, number 2, so you would remove that cutting. And 3, the cutting was placed in a container with soil. So depends on where you have your nursery. So in this case, in this diagram, that cutting was prepared and placed into a container with soil, and it will be left there to grow and develop into a new plant. Of course, these methods, I'll do different individual videos for them to demonstrate how they are done. There are more information relating to how they are done, but that will not be explained in this video. I have already done circumposing in another video on the channel, but I'll also do other demonstration of the different methods. So look out for those. So keep in tune, alright? Remember to subscribe to the channel to get those automatic updates. Let's move to layering. So we completed cutting. Let's move to layering. So layering. This process involves making a stem growing roots while it is still attached to its parent plant. Alright, so you have a plant, you can make the stem, a stem of the plant, grow roots. But you would not remove the stem. You would allow the stem to grow roots first, then you will remove that stem after, with the root system and a shoot system. Alright, so with cutting, you remove the stem, then you set it to grow roots. But with layering, you allow the stem to grow roots while that stem is still attached to the parent plant. Alright, so example that this method is carried out on is like your hibiscus, your croton, your ficus, your cherry, your roses, etc. Alright, there are 2 types of layering. Number 1, you have ear layering, and number 2, you have ground layering. Alright, with ear layering, ear layering is oftentimes called circumposing or marcotting. So those are 2 other names for ear layering. Alright, with ear layering, the stem rooting stays above the ground. Alright, so the stem that you will make root, so this is where man is involved also, is man will carry out that procedure. Alright, so the stem that is rooting will stay above the ground. Alright, so you'll take soil medium to that stem. For example, your sawdust, your peat moss, you will take the rooting medium to that stem. You will not allow that stem to be in the soil. Alright, so with ground layering, the stem roots in the soil. So in this case, you will bend the stem and peg it in the soil to grow roots. So with air layering, it's above the ground. With ground layering, it is in the ground. This is a diagram showing you air layering. So of course, this yellow part here is the soil. So this is the plant, and of course, this is the circumposed area above the soil. So this yellow structure here is the soil. So with this area here, the sawdust or the peat moss is added here for the stem to grow the roots. Notice not in the soil. Alright, so this is a diagram showing ground layering. So this is a plant here. The yellow structure is the soil. So please note that the stem that you want to grow roots, it is pegged in the soil. And the roots will grow right here. Alright, so that's the difference between the two types of layering. Alright, so let's move to budding. Alright, budding is the removal of a single bud. Now that bud that you remove from a plant is called your scion from a plant and putting it on another plant to grow. So the second plant that you put it on to grow, that's called your rootstock, or for short, you can say stock. Alright, so the plants must be of the same species. So with this method, those two plants must be of the same species. So of course, if you have a blacky mango and a Julie mango tree and you want, say you have a blacky mango tree but you want Julie to grow on the blacky mango tree, you can take your scion from your Julie mango tree and you bud it onto your blacky mango tree. Alright, so they have to be of same species. So mango to mango, citrus to citrus. Because the DNA of the plants they will match. So the DNA of mango tree will not match with the DNA makeup of the citrus plant. So that is why you will not be able to cross them to different varieties of species of plants. Types of budding methods. So here we will discuss two types. One, the inverted T method, and two, the patch budding. Alright, so let's move to the next slide. Alright, so here you have the inverted T method. Alright, so one, you would select your stem from a plant. So as I said, so one scion or bud removed from a plant. Alright, so in the example that I was using now, say your Julie mango tree, you would remove a bud. Now you need a little bit of the bark and wood onto this bud. So this is the bud, this is the black area here, is the bark with a little piece of the wood. Alright, and that would be about maybe an inch long, etc. So number two, now you would select the plant that you want to put it on, which you call at your stock. So for example, this is the blacky mango tree here, right? So you would select a stem on the blacky mango tree and you will make an inverted T-cut into the bark. The reason why you make this inverted T-cut is so that you can lift the bark. So you could put the scion inside. Alright, so number three, insert bud scion into the inverted T-cut. So you'd use your budding knife to lift the bark and you insert that scion inside, leaving out the bud. And number four, secure the bud using plastic tape to hold the bud in place and to prevent water from getting into the T-cut. So of course, you would use tape to secure or to wrap around the bud to seal up the T-cut, alright, to hold the bud in place. Alright, so that's basically how budding the inverted T method is carried out. So you will leave this plant to grow and watch it to see if the bud will develop. In some cases, what you can do is to remove the other vegetative parts of the stalk to help the nutrients to go to this bud. The patch budding is different. In this case, alright, so one, a patch of bark and bud removed from the scion. So you have your plant, say you can also use this on the Julie mango tree. Say you remove a bud, but in this case, it would look like a shield or a square, a rectangle. So you remove a rectangle of bark and wood with the bud. Alright, so that's the main difference. Number two, you will select your stock. Say the blacky mango tree, you select a stem on it and you use your budding knife to cut out a rectangle in the bark, similar to what you have done with the scion. Alright, number three, you insert the scion into the stock, which is here, right? And then, of course, four, you would wrap with tape or secure with tape. And you will leave to develop. So the bud will develop. So you would observe, and as I said, if you want more nutrients to go to this bud, what you do is to cut off the other stems from the stock plant. Grafting. Alright, grafting is the removal of a stem with a number of buds. We call that the budwood from a plant and putting it on another plant, which is the stock. Plant should be of the same species. Alright, so with budding you remove a single bud. With grafting, you will remove a stem from the plant and that stem will contains a number of buds. Alright, so of course you have different methods. So you have types of grafting methods. So we have 3 of them that I'll explain. You have the cleft grafting, 2 you have the veneer grafting, and 3 you have approach grafting. Alright, so let's move on. Alright, so here we have the cleft grafting. Alright, so number 1, you would of course select your scion. So you have your Julie mango tree. So we're using that example throughout. Alright, so you have your Julie mango tree, and you want that Julie on your Blackie mango tree. Alright, you want a plant of that Julie, so you're putting it onto the Blackie. Alright, what happened now? You would select a stem, and remember the stem will have more than 1 buds on it. So you select a stem, you make a wedge cut on that stem. Alright, this can be about 5 inches tall. Alright, number 2, you select your stock, which as I said is the Blackie mango tree. Number 3, you make a V cut in the stock. Alright, so you would actually make a V cut, which is here in the stock. Alright, so this V cut here, it should match this wedge up here. Alright, and then 4, you insert the scion into the stock and wrap with tape. So of course here, you see where the scion now is inserted into the V cut. And then you would secure with your tape and observe for growth. Alright, so that's basically the cleft method. Just observe that the wedge is the wedge that makes the difference. Alright, with the veneer grafting, you would not make a normal wedge. So with 1, you select your scion, as I said earlier, but it would be like an irregular wedge. Alright, so please note, it would be about 5 inches tall, etc. Alright, so number 2, of course, you would select your stock, which is your Blackie mango tree. 3, you would make a irregular V cut in the stock. Alright, so you'll cut your stock here to match the scion, the shape of the scion up here. And then what you do now, you would fasten them, you would insert them together, and then wrap and leave and observe for growth. Alright, so it's just shape that makes the difference with the cleft and the veneer. And with the veneer, it can be done on the side of a stem. But with the cleft, it is done on the top of a stem. Alright. Alright, the approach grafting and the last method of grafting. Of course, 1, you would select a stem from a plant and remove a piece of the bark. So, alright, so this plant here that is in the soil, say for example, this is your rootstock. This is your Blackie mango tree. Alright. Now you want the Julie mango tree onto this Blackie mango tree. Then what you do is to, of course, select a stem on your Blackie mango tree. Cut off a piece of the stem, just scrape off a piece of the bark for joining onto the Julie. So of course now, if you notice, you'd have a second plant, which is your Julie, which is number 2 here. Select a stem from plant 2 and remove a piece of the bark. So you would not remove the stem from the plant here. Alright, so this plant is in a pot or a container. So you'd bring the plant up close where you could join the 2 stems, and you cut both stems and join 2 cuts together and wrap with a tape. So number 3, bind the 2 cut stem together using tape and leave until they become 1. So what they will do that is that the Foley vessels of both will join together. So that's the reason of cutting them. So they will join together, and you observe once they catch together. Then number 4, when the stem fully merge as one, cut off the scion using a secateur. So after they merge, you will cut this stem right here. You'll cut it off, and then this Julie mango stem will be left onto the Blackie mango. Alright, so let's look at this diagram over here. So eventually this stem here will be left onto the Blackie mango tree, and of course the stem to grow, you can remove the other stems from the Blackie mango tree. Now when I said Blackie mango tree, the size, it could be a small Blackie tree, it could be a very small one. Alright, so you could remove the stems. It could also be a large tree, but you'd have to help remove the other parts to help this new stem to develop. Tissue. Let's go to the definition. So tissue is the removal of a piece of plant, normally a tissue, a very small part, from the leaf, the stem, root, and growing it in a solution of agar, nutrients, and hormones. And you normally carry out that in a lab. Alright, so with tissue as I said, tissue, you remove a tissue from the plant. Tissue is a very tiny part of the plant, so you remove a piece of tissue. So a tissue will have a number of cells. Alright, so a very small piece of plant that is removed and cultured to grow in a lab. Alright, so let's take a look at this diagram. Alright, so here you'd have your container with, could be a glass, a petri dish, or a glass here, with your solution, and of course the plant tissue in the solution. This is normally done with banana plants. And of course, you would observe until it start growing. So over here, in this diagram here, you see where the plant is starting to grow leaves and roots. So of course, this procedure is done in a lab in a controlled area where the procedures will be observed daily or regularly. Alright, so let's move to the advantages and disadvantages of vegetative propagation. Alright, after this we'll move into the summary of the lesson, which will be very short. Alright, so number one, let's look at the good thing about this method. Alright, number one, so offsprings are identical to the parents. Alright, so you have some plants and you want exact offsprings, then this method is good. So you have a nice mango tree or a nice orange tree, sweet, then this is the method to use, because if you use sexual, the offspring may not become sweet. So if, for example, you have a sweet orange tree and you use sexual reproduction, when those seeds grow into new plants, they might not bear sweet orange because the father that contributes, that donates the pollen grain, that might be a sour orange tree. Alright, the new offspring might come sour. Alright, so number two, new plants can bear fruits in a short time. So when you use these methods, as I said, from seed plants might take 5 years like a mango, or 4 years. With this method, within 2.5 years, 3 years, it can be ready. Alright, reach mature stage and growing. And bearing. Number three, reproduction is fast. Alright, so you can use this method to produce a lot of plants in a short space of time. So if you have a farm with hibiscus and you want to produce like 500 in 1 week, that can be done very easily. Alright, let's look at the disadvantages of vegetative propagation. So these are the bad things about it. Alright, one, diversity is lost in asexual reproduction. So it means you should not just practice to carry out this method all the time. Alright, because if you just practice this method, then you will lose diversity, variation, the chance of developing a new variety. Alright, so number two, new variety of crops cannot be developed in this type of propagation. Three, require special skills to perform. Alright, so with this you have to do training and exposed to how it is done in order to do it. As I said, I will do videos carrying out demonstrating all these methods of asexual reproduction, all the artificial methods. Alright, the summary of the lesson. Alright, so here we have the major topic, which is reproduction. We said there are two types. So here we have sexual and over here we have asexual. Now, the sexual would involve sex cells that are produced by the flower of the plant, and they normally will produce seeds, and when you plant those seeds, of course you get new plants. With asexual methods, this does not involve the use of seeds or flowers. Alright, you have two types. So in other words, it involves the leaves, the stem, the root, the buds of the plant, all the vegetative parts. Alright, you have two types. You have natural and you have artificial. With natural, no man is not involved in inducing the reproduction. So the plants normally carry out these methods. They use different plant parts. So like the bulb, which is used by the onion, you have the rhizome, which is by the ginger, corm. So this should not be corn, it should be corm, C-O-R-M. You have stem tube or root tube or suckers. Alright, with artificial methods, you have cuttings, layering, budding, grafting, tissue culture. Of course, with the natural, man is not involved in aiding the reproduction, but with the artificial method, man is involved in aiding the reproduction by carrying out different techniques. Alright, so we are now at the end of this lecture. Please remember to like and share the video. Also, if there's something you don't understand, please leave a comment. I'll try my best to give a response. Thank you. Until next time. Please remember to subscribe to the channel.